So thank you. This is my first time in one of these Leman's conferences, and I thank Monse and Marie for inviting me. I'm a little bit embarrassed when people mention that I'm a chair in poultry medicine. I'm not a veterinarian. I'm a biochemist by training. So for the biochemist, I became more like a poultry person. For the poultry people, I'm still a biochemist. So I don't know what I am anymore. The one thing that I do notice is that you are taping this, and I will ask you not to put it on social media because my wife doesn't know that I'm here. So, I will go ahead into the brief introduction. The topic of my interest is influenza viruses. Within these type of viruses in the Orthomyxoviridae family, there is one type called type A, influenza viruses. And these viruses, what they have in common is a segmented genome of RNA of negative sense. And in the case of type A influenza virus, there are 8 segments. And there is a large reservoir of these viruses in aquatic birds of the world. And from this reservoir, it's thought that in many instances, strains emerge with the ability to jump into other species and cause disease outbreaks. Based on the 2 major surface glycoproteins of these viruses, hemagglutinin and neuraminidase, there are 18 hemagglutinin subtypes and 11 neuraminidase subtypes, and they can happen in many different combinations in the wild bird reservoir. But when it comes to jumping to other species, the number of HA and A combinations that we see is much more limited. And these viruses, as you know, have been responsible for pandemics in humans through a process called antigenic shift. That is when a new subtype enters into a naive population, in this case a human population. And many of you have heard about the 1918 Spanish flu. One of the most devastating pandemics in human history was caused by influenza A viruses. And more recently, we have the emergence of a new pandemic strain, the 2009 H1N1 pandemic virus that originated in pigs. And these viruses are so-called triple reassortant viruses because they have gene segments derived from evolutionary history related to gene segments from the avian reservoir, swine, adapted influenza viruses, and human influenza viruses. But there is also a two-way transmission that is happening constantly. And in fact, there are many more human viruses that go into pigs and get established in pigs than the other way around. And that has increased the diversity of influenza viruses in the pig population. To the point that makes it very difficult to make vaccines against these viruses because it's very important to know what is out there in order to have a vaccine that works. In addition, influenza viruses are known to evade the immune system very easily by incorporating point mutations, particularly on the hemagglutinin protein, which is the major target of the immune system. And the more red you see on this, the structure of the hemagglutinin, the more susceptible to change that position is. And this is a way for the virus to evade the immune system, and in that way we can get reinfected, pigs can get reinfected, or vaccines become not effective. So we are in this constant race in trying to understand influenza viruses, what they look like on the surface, and how to make a better vaccine for it. So in the case of swine, we have the diagnostics laboratories providing information. Eventually, to understand how these viruses move, we have sequencing technologies. But ultimately, we rely on, I don't know if you can see the pointer, it's very weak, but in an antigenic characterization of these viruses. What these viruses look like on the surface is very important to have a vaccine that works against them. And usually these hemagglutination inhibition assays, which is a typical assay that is used to characterize these viruses, are really developed into big tables that are difficult to understand. So back in 2004, Smith and later Bjorn Kohl in 2013 developed an assay which is, or not an assay, but a system to visualize these hemagglutinin large data sets. And that's called antigenic cartography. And essentially what you do is to put each one of your virus strains on a map, and that tells you how related each strain is to each other. And the work that Bjorn did basically revealed that what controls or what modulates HI activity is related to a few amino acids on the surface of the hemagglutinin that are very close to the receptor binding site of the virus. And perhaps luckily for us, then Nicola Lewis in Cambridge also noticed that pigs see essentially the same thing. The antibody response of the pigs see the same thing on the hemagglutinin of swine H3 influenza viruses. Few amino acids are involved in this modulation of HI activity. And we, in collaboration with Amy Vincent and NADC, could actually show that we can modulate this activity by introducing mutations in the genome of the virus, in the hemagglutinin. For example, if you run an HI assay, you have this cluster here, and these amino acids here, you can establish motifs. So these motifs are these amino acids here. Each one of these corresponds to a position in this motif. And you can have HI activity against a cluster of viruses like this, the CN cluster. And then if they accumulate some mutations here, there are few amino acid mutations, you change the HI activity of these viruses. And you can actually recapitulate that by incorporating those mutations. So if we take this Ohio 04 virus with this antigenic motif here and make it more like this red cluster, now the HI activity of this virus becomes more like this virus. And the same goes true for another virus called of the green cluster, in which this motif here is mutated to this motif in here, and now your virus behaves like the green virus. But when we analyze what circulates in pigs, it is really complicated, because there have been many introduction of H3N2 human-like influenza viruses into pigs, in these cases in North America, and then you can have many viruses circulating with many different antigenic motifs. And since this is very important for receptor recognition, for the antibody response, and vaccines rely on the HI assay to predict whether they are going to work or not, getting a vaccine against influenza in pigs becomes a real challenge to really match antigenically what circulates out there. So in terms of your building blocks, your favorite Legos to make a vaccine for influenza, there are many different technologies. The traditional technology is the whole inactivated influenza viruses that most typical they would have some sort of adjuvant, and they can generate a good HI response. They can generate a strong hemagglutination inhibition response, mostly to that homologous strain. But then if you have a whole inactivated vaccine against the homologous virus, usually these vaccines work very well. But if there is a drifted variant, so again, in this case would be a variant that has a different motifs from the vaccine strain in that region that I just showed you, then your vaccine is less efficient. And you can also have some other recombinant vaccine strategies that usually relied on cloning a hemagglutinin gene into the vector of choice and vaccinating animals with that vector of choice. Again, you're eliciting an immune response against the hemagglutinin. Whole inactivated vaccines tend to produce a response against the hemagglutinin more prominently. And then we have live attenuated vaccines, and these are, I'm biased because we work with live attenuated vaccines, so I prefer to see live attenuated vaccines being used out there. Because live attenuated vaccines, unlike any other technology, can mimic a natural infection. So when you use a live vaccine, you're mounting a humoral and cellular response that most closely resembles what a natural infection would do. So the immune response becomes multidimensional. Even though you want antibodies against the hemagglutinin, you're also generating a response against many other components of the virus that, if you have good immunity against that, can prevent the virus from replicating or transmitting. So in our laboratory, we developed a live attenuated vaccine strategy that you can also discriminate the vaccine from the wild-type virus by using a PCR. And in this case, the virus contains one of the gene segments of the virus, one of the polymerases, 3 amino acid mutations that give a temperature-sensitive phenotype, as well as another mutation in one other polymerase subunits. And we added an epitope tag that is also derived from influenza at the carboxy terminus of PD1. And these viruses are very stable. They tolerate multi-passages in eggs or tissue cultures without losing this temperature-sensitive profile. And that can be measured in a polymerase assay by looking at different temperatures. The virus can show polymerase activity at low temperatures, but compared to a wild type, start losing polymerase activity as you increase the temperature. So we have published extensively on this strategy. We recently used some of these mutations to make an influenza B that affects humans, also temperature sensitive. So we have a type A and a type B live attenuated vaccine. And recently we published a quadrivalent vaccine formulation that shows a promise as a quadrivalent vaccine. Obviously, we didn't do this directly in humans. We use mice. As I always say, those of us working in the vaccine world have saved the lives of many mice around the world. Still remains to be seen whether they will be useful to humans. But then we show that at least it has the potential. Now, when it comes to pigs, we have published extensively on this, and the latest, which has just come out in Journal of Virology, is a study in which we did 2 separate studies, in which in the first study we had pigs of 3 weeks old that were primed with a live attenuated vaccine that belongs to the cyan cluster. And then they were challenged, they were boosted 5 weeks, and then challenged 3 weeks later with a red cluster vaccine virus. And they were compared with a similar formulation of an inactivated vaccine, adjuvanted vaccine. Okay? In the second study, we have older pigs. I believe those were 9-week-olds when we vaccinated them. They were boosted at 11 weeks, and then challenge 3 weeks later with either a homologous challenge, so the same as in the vaccine, or a antigenically drifted variant. In the case of the vaccine here, this is a green cluster surface genes in the background of the cyan cluster, this Turkey 04 or Ohio 04 ATT virus that I showed you in the previous slide. And different studies have different number of pigs. And we tested for protection against pathology and challenge. So what we're talking about here is that we vaccinated with this virus here and challenged with a green cluster, with a red cluster virus, or we vaccinated with a green cluster virus and challenged with a red virus or the homologous virus. When you look at HI activity, you will realize that there is very little cross-reactivity, almost none. These are the homologous HI titers. And if you look at the table carefully, you will realize that the different viruses show much reduced HI activity with the drifted variants. So when we look at what happens after challenge in the first study, what we see is that there was not much in the way of lung lesions, but obviously those that were not vaccinated, those that were non-vaccinated and challenged show some of the more prominent macroscopic and microscopic lesions, followed by the pigs that received the inactivated adjuvanted vaccine. And there was, with the live attenuated vaccine pigs, there was the type of lesions that we saw were almost indistinguishable from those that were not, the pigs that were not challenged. And in the case of the pigs that received the live vaccine, that was the only instance in which we could observe sterilizing immunity, both in the bronchoalveolar lavages at 5 days post-challenge or in the nasal swabs. Now, one of the things that has been described by the Amy Vincent's group is that depending on your antigenic mismatch, if you use a whole inactivated vaccine with an adjuvant and it's not a perfect match, you can lead to a vaccine-associated enhancement of respiratory disease. And that's what we see if we use the Iowa 14 whole inactivated vaccine and challenge with the New York 11. But you don't see that if you use a live vaccine. You have very nice protection from microscopic or microscopic lesions against challenge. And once again, with the live vaccine, that was the only instance in which we could see sterilizing immunity. Now, sterilizing immunity may be utopic to achieve in the field, but when it comes down to vaccines, you want to use the best vaccine you can. Okay, so this sterilizing immunity that you see here experimentally may not happen in the field, but why would you go with a vaccine that is less efficacious even experimentally? Okay, so that's why we tend to favor the live vaccines. Another thing that is important is that, and this is just one example of many, your levels of antibody responses by HI in the serum do not necessarily predict how much shedding you're going to have after challenge. Okay, you can have very high HI titers, like in this case with the whole inactivated Ohio 4 vaccine, you have very high HI titers. But as I showed you in the previous slides, still after challenge there was significant amount of virus shedding. And with live vaccines, you can have very little in the way of immune responses, but still have complete protection. And that's probably because most of the actions of the live vaccine is at the mucosal level, okay, where you have both the elicitation of IgA and IgG responses at the mucosal level, and they tend to be more cross-reactive at that site. Now, is the live vaccine, the ultimate vaccine? No, it is not. In a previous study that actually I like very much, was done in 2013. This is just one slide about it. We use 2 live vaccines. This live attenuated vaccine 2 is ours. This is Ohio 04 ATT. This is another type of live attenuated vaccine, which is antigenically significantly drifted from the challenge virus. And what you have is that with our live vaccine, we have no virus shedding, but pigs that were vaccinated and challenged with the other live vaccine, they show some levels of virus shedding. In this experiment, the thing I like about it is that we put pigs that were vaccinated and challenged, and then we put respiratory contacts, indirect contact pigs, to see whether any virus that was being shed by the vaccinated and challenged pigs was going to be enough to start a new chain of transmission. And the thing is very simple. If you have no virus shedding from the live, from the vaccinated pigs, there is no transmission of a live virus, is that right? But any pig in the other groups that was shedding virus, that was enough to start a chain of transmission in the indirect pigs. And there is an inverse correlation based on the levels of virus shedding and when transmission occur. So that highlights that even though a live vaccine may be better than any other vaccine, still you will have the dilemma of trying to come up with antigenically closely related strains in order to vaccinate. So that's why there is a big emphasis in trying to develop universal vaccines. And universal vaccines, in essence, are strategies that are aimed at eliciting an immune response against less variable epitopes on the virus. And there are many proteins in the virus that have less variable epitopes, including the hemagglutinin. You can target regions of the head of the hemagglutinin that is less variable. You can try to target the stem of the hemagglutinin stock that is less variable. Antibodies against the neuraminidase are also important in protection. May not be as important for initial infection, but they certainly reduce virus shedding. And then there are other proteins that are more stable, like less variable, like the M2, also on the surface of the virus. Antibodies against the M2 have the potential to protect. I think we have some ways to go with universal vaccines. It's not that easy to overcome a virus that has evolved strategies to actually fool the immune system all the time. And then we are also looking into universal platforms, platforms that can be applied indistinguishably in many animal species. So if you think about a universal platform that we have been using for a long time, not necessarily that the virus is the same, but whole inactivated vaccines, you can see them as a whole as a universal platform. We have been using whole inactivated vaccines to prevent influenza in people, influenza in horses, influenza in pigs, influenza in avian species. But certainly they have many drawbacks. And one of the major drawbacks is that they only they are not very good at eliciting a mucosal immunity, which I think is crucial to prevent influenza. And then you have live attenuated influenza vaccines. There are different strategies. One of them is already approved for use in pigs and is commercially available. There are vector vaccines. There are nucleic acid-based vaccines or alpha virus replicons that are also approved commercially. And then you have these subunit vaccines. All of them will have to address that there is a significant amount of antigenic diversity of influenza viruses in pigs that make the selection of vaccine candidates difficult. I think that we need to better understand what contributes to the antigenicity of influenza viruses. And we have to develop systems that can help us better predict what an amino acid change means in terms of HI activity. We're getting very close. As I show you, there is an antigenic motif that seems to modulate HI activity. One of the things that I did not mention is that even though influenza can change all the time, at every position, not everything goes. So flu can mutate all the time, but the number of amino acids that change in each position is not for whatever amino acid exists in nature. There is a limited range, and flu tends to recycle amino acids, something that in my lab we call it amino acid recycling. I don't know if that's the right term, but flu does this a lot all the time. The problem is with the combination of amino acids. So the combinations are not always the same, but at each position you may have recycling of an amino acid at any given time. Live vaccines, the ones that we have used so far, you don't see there. You don't see this enhancement of respiratory disease, unlike you see with whole inactivated vaccines. Whether any recombinant vaccine out there would also have the same effects as a live vaccine with no VAER or as an inactivated vaccine with VAER, it remains to be seen. I don't think it has been explored in detail. And one issue that comes up often in questions is that can live attenuated vaccines contribute to influenza diversity via reassortment? And those of you working with pigs can probably answer this much better than anybody else. can you make things even worse than what they are right now in terms of the antigenic diversity or virus diversity of influenza in pigs? I will stop here. I have to thank the Vincent Lab in NADC. None of the work that we do in pigs with live vaccine would have been possible without her help. And past and present members of my lab and the funders for providing the resources for us to do the research. Thank you.